Hey there, welcome to Beauty at Work. In July 2022, we held an interdisciplinary panel at Magdalen College at the University of Oxford on the role of beauty in science. This event was sponsored by Templeton Religion Trust and Magdalen College. The panel represented perspectives from diverse disciplines: mathematics, physics, biology, philosophy of science, and sociology. To see the slides from the panelists' presentations, please see the show notes or the link to the YouTube video. This is the second episode in the series. In our last episode, we heard from Dr. Ben MacArthur and Dr. James McAllister. In this clip, we're going to hear from three other panelists. Dr. Milena Ivanova is Bi-Fellow at Fitzwilliam College, the University of Cambridge, and co-editor of The Aesthetics of Science, author of Duhem and Holism and The Aesthetic Nature of Scientific Experiments. Dr. Robert Gilbert is professor of biophysics in the Nuffield Department of Medicine and director of the University of Oxford's Medical Sciences Graduate School. He's also author of Science and the Truthfulness of Beauty. Finally, Dr. Sabine Hossenfelder is the author of Lost in Math, How Beauty Leads Physics Astray, and Existential Physics, A Scientist's Guide to Life's Biggest Questions. And she's also creative director of the YouTube channel Science Without the Gobbledygook.
Thank you so much, everyone who is watching online, who is in Oxford. Thank you so much to Brandon for inviting me to be part of this panel. It's a great honor to be in such amazing company. Very sorry not to be there in person. COVID was a lot more of a struggle than I expected, but I'm very pleased to be able to still join the conversation and explore the aesthetics of scientific experiments. So I'm interested in understanding whether experiments in science can be objects of aesthetic appreciation. And the questions that we're going to think about today is whether we can appreciate an experiment for aesthetic reasons, whether we can find, for instance, an experiment beautiful. What would be the focus of this appreciation? What would it mean to say that an experiment is beautiful? What would we actually focus on? And whether there is any stability in how we appreciate aesthetically. This is exactly leading from where James just left us, arguing that there is dynamicity in how we understand beauty in science, and we see these continuities in science. So how is this to be understood when we think about experimental practice and how much it has changed historically? So I'm going to start by presenting this distinction here. Well, many ways in which we can appreciate aesthetically an experiment. We can appreciate the phenomena that the experiment reveals to us, the instruments that we are using. We can also think about the performance of the experiment itself as something we appreciate aesthetically. This is particularly prominent in earlier experimental traditions where the experiment was practiced as a performance in front of audiences. We also appreciate design of an experiment, and that will be really important to my argument. The very process of conceptualizing a design and a setup for an experiment is also subject to aesthetic judgment and can be appreciated aesthetically. And finally, the results. So let me just give a few examples of all this to illustrate it. So first, the phenomena. This is just a few examples of the kind of amazing things you can see under a microscope if you're in the lab and doing an experiment, but also the kind of instruments you can see if you are, well, depending on which kind of experimental tradition you're part of. The first to the left and to the right, the third picture, are from some instruments that we house here in the University of Cambridge in the History and Philosophy of Science department. And they're very clearly, aesthetically very pleasing to look at. They used to be, of course, used. And the experiment in the middle is the pitch drop experiment, which I used to pass by every day on the way to work at the University of Queensland. And that's one of the kind of very public experiments. And it resembles very much the kind of performative experiments that used to be more part of scientific practice. But we can see that the instruments themselves can be very pleasing. And the performance of the experiment can be very much analogous to artistic performance. And here in this famous painting by Joseph Wright of Derby, this is very well illustrated. This is an experiment that was performed with a new instrument, the air pump. And in the early days of the Royal Society, the experiment used to be very much a focal point of appreciation for aesthetic reasons. So it will have audiences where people engage with the experiment and appreciate it, and often appreciate nature as it's revealed to them. And this painting is brilliant because it shows us how many different ways people appreciated aesthetically the experiment, from awe and fascination to even fear and all of the kind of the diversity of aesthetic responses that would have to the fact that this bird is going to die because we're extracting the oxygen from the cylinder. So the performance is aesthetically appreciated here, and the experimenter is like an artist in the middle of the painting. But what I have argued in some of my work recently is that these features are kind of like a nice bonus to have, right? So they're visual features that are very kind of easily perceivable to us, but they're not necessarily sufficient to make an experiment aesthetically valuable or to make it beautiful. We're going to talk more about beauty today. What really matters is this very special relationship between how the design delivers a result and the kind of imagination and creativity, ingenuity that is involved in conceiving such an experimental design. So let me illustrate this a little bit more. In one of these famous experiments, Foucault's pendulum, we see this really well illustrated. It was an experiment that clearly had some very visually pleasing features. It was basically a brass ball that was hung from a rope in Paris's Pantheon, and people would go to see it. As you can see here, there was always an audience admiring it. Beautiful pendulum swinging around. But what's beautiful about this experiment has nothing to do with that. I mean, okay, very nice that we can see something visually pleasing, but what is really ingenious about it is how simple the experimental design is for the purpose that it had and the significance that it had. The significance is really important. This experiment with this very simple setup shows something very previously disputed, and that was that the Earth rotates around its axis. And the experiment delivered that by simply showing that after some time of this pendulum swinging, the sand underneath, the lines that the ball had made, were not aligned with each other. So really important results delivered by a very elegant, very simple design. And this is what I think makes this experiment beautiful. The visual features are kind of secondary to this much more substantial beauty that we find in how the results are delivered by a very, very good design for purpose. Similarly, in biology, we celebrate the Meson-Stowe experiment because it delivered important results to a really, really important question, and it delivered them in a really ingenious way. So this experiment has received the title of the most beautiful experiment in biology for good reason. As historian Hans-Peter Fischer has argued, there was something really ingenious about how the experimental design was basically set up. Beautiful idea to use basically the method of centrifugation, to use density to study the genetic material through the generations as they did in order to determine how it changed. And there was some simplicity, as historian Frederick Holmes argues, in this setup, in the steps that they followed. So simple, elegant design, but the results that the experiment delivered were also incredibly significant. James Watson, one of the discoverers of the double helix structure of DNA, argued that this experiment was ingenious because it gave a simple answer to a question. We treat this experiment as an example of a crucial experiment in science because it delivers these really important, straightforward, easy results. Right? It's not a messy result. It's really quite clear, speaking in favor of one of the three contenders at the time of how DNA would replicate. Semi-conservatively, the answer was. And Peter Fischer argues against that the experimental results just spoke for themselves. There was no need to replicate them. No further commentary was necessary. And Holmes argued that there was this immediateness about them, that there was a singular historic event. We didn't have to worry to redo these experiments many times. Of course, recognizing that that's quite unusual, but that was one of the aspects that was really valued about how the experiment delivered its results. And what's worthwhile noting here is that so far we have these two experiments as examples of beautiful experiments that clearly delivered an answer to a question and delivered an answer to a question that aligns with expectation. Right, in the first case, we have a demonstration of a phenomenon that was expected. And here we had the selection of a hypothesis among three contenders at the time, aligning against the expectation. Sometimes things don't go like that, though. In science, we have sometimes experimental results that will not align with expectations. So the question is, what happens there? Are they then making the experiment aesthetically disvalued? We can think about the Michelson-Morley experiment as an example of something like this and how it has been praised by scientists as a very beautiful experiment. Albert Einstein himself argued that Michelson was an artist in science, the artist in science, right? Really highly praised for his aesthetic sensibility, who took great pleasure and great joy in the beauty of the experimental setup and the elegance of the method that he employed. And historian Gerald Holton argued again about the ingenuity of just setting up the experiment and building the instruments necessary to achieve the goal of the experiment. Nobody before Michelson was able to imagine and construct an apparatus to measure the second-order effects of the presumed ether drift. He argues that the interferometer was a lovely thing. So what happens in these cases where we have results that don't necessarily align quite with expectation, but are really well set up for purpose? I think we should understand them still as beautiful, but understand the nature of significance in a slightly different way. So by significance of results, we can think about them as going our way when we have a demonstration of a phenomenon or confirmation of discovery. But also sometimes we have these disruptive results that are really productive. Not every null result will be like that. But sometimes, like in this case with the ether, not detecting the ether, actually was what was significant and what was really good about this result, this experiment. And so I've argued that we should really think about this kind of productive disruption as part of being a significant result that makes an experiment really beautiful, but the kind of significance it had in revising beliefs, opening conversation, and reconsidering fundamental assumptions that are being made. If an experiment can do something like that, that also should be considered as part of being significant and well made for purpose. So, framework I'm suggesting here argues for the following things: that experiments can elicit aesthetic responses. These can be very pleasing, but sometimes disturbing as well. And that's very analogous to how we appreciate artworks. Not every appreciation of an artwork is pleasing or is simply appreciation of beauty as something that's very easy to process. Sometimes they are disruptive as well. I've argued that the visual features are really nice to have sometimes. They're not essential for us appreciating an experiment as beautiful. The beauty judgments really pertain to how the design and the significance of the results are intertwined and how they reveal ingenuity in the setup of this kind of appropriate design for purpose. And I think I want to kind of go back to what James was arguing just 10 minutes before about the fact that we can change the way we experience beauty in science. Clearly, experimental results and experimental setups today are very different. How we get to our results is very different. We use machines. The setups are very different, very big machines, very big detectors. Nothing to do with the 17th century experimental setups that we see, the tabletop experiments. The experiments look so different. They're practiced so differently. So how can we talk then about having some kind of notion that employs to all of these particular setups? I think thinking about beauty as a functional notion will help to see that we can still praise that kind of relationship between design for purpose, even if we recognize that the setups are very different. So how will we actually materialize a really good design for purpose will definitely be changing depending on the kind of area that we're studying and the kind of setup that is available to us. But this, I think, can maybe unify the different ways in which we practice experimentally. So this is from me. Thank you very much for your attention, and I look forward to the rest of the talks.
Thank you, Brandon, and it's lovely to be here. As Brandon says, I'm going to talk about nature and data and beauty in those. And for me, as also for colleagues that I work with, nature and data come together under the topic of biophysics. And what I'm going to say, happily, is going to follow on nicely from the previous three presenters. So, and in terms of what I want to talk about, I'm actually going to group the points I want to make, which, as I say, come very much from my personal point of view, under a series of more general terms, which you will find used in all sorts of other kinds of discourse, because I think they are helpful ways to think about the kinds of applications of perceptions of beauty that are going on in these different areas of science. And the first thing I'm going to talk about is a couple of examples of idealism. This also happily follows on from Milena, because this actually relates to the Mieselson style experiment that she was talking about. So on the left-hand side of the screen, you see a famous cartoon drawn by Watson and Crick for their paper published in 1953 describing the structure of DNA. And if you read that paper, a very short paper, which you'll be able to find on the Nature website, I'm sure perhaps you've all read it, you will remember if you've read it, or you'll find if you go and read it, that it's framed very much in ideal terms. It's very much about what would be neat given the data we've got. There were certain bits of information they had, but they needed to put them together into a satisfying whole, and they came up with the double helix hypothesis, which turned out to be entirely correct, essentially, and which leads on, of course, on the right-hand side to the experimentally determined structures for DNA itself, and of the more complicated RNA, which has because of a particular chemical modification in ribose compared to deoxyribose, has a much more complex range of folding patterns. But at the core idea that Watson and Crick came up with was correct, that base pairing leads to the double helix. The reason it relates to Milena's talk and to the mesos and the Stahl experiment is that, again, very famously at the end of the 1953 paper, and sort of with their tongue in their cheek, Watson and Crick said, "It has not escaped our notice that this structure suggests a possible mechanism for the replication of DNA." And of course, their mechanism was exactly the mechanism which Mesos and Stahl went on to show was correct, the semi-conservative hypothesis for DNA replication. So that was an idealistic process. Idealism 2 here is another example of, which is, in this case, relates to symmetry, I guess, as does the DNA example. Also, Watson and Crick, in 1956, published again in Nature a paper describing what they thought the structures of small viruses would be like. And they needed to solve the problem of how it is that you can enclose a genome with a protein that it's encoded. And this is a basic problem to do with the size of the molecules. And they came up with some simple ideas about how you could essentially organize triangles in order to generate closed structures which would enclose the genomes of viruses. They did it perhaps informed by knowledge about the Platonic solids, which you can see some ancient dice down below replicating those, the 5 Platonic solids, of which the one which gets the most complex structure with the simplest shape is the icosahedron. And that was indeed what they proposed would be the structure of small viruses. And this is essentially as a theoretical paper by Watson and Crick, which turns out when you solve the structure of viruses like the blue tongue virus or any number of other isometric viruses, and this was a paper published, as you can see, 25 years ago or so by some colleagues of mine here in Oxford, the blue tongue virus, 100 million Daltons, the largest structure to be solved at the time. 720 copies of one protein forming outer shell, 120 copies of another protein forming an inner shell, the DNA, the double-stranded RNA of the genome also being resolved. It essentially has this icosahedral symmetry, and so their theory is correct. But of course, all symmetry is not perfectly isometric, and so you can also play with the same ideas to come up with something which is more distorted, but nevertheless has the same core idealistic senses behind it. And in fact, the structure of the HIV capsid turns out to use that idea. It also, like icosahedra of an isometric form, has 12 pentagonal vertices, but they're arranged in a different set of positions to where you would find those 12 pentagonal vertices in blue tongue virus, where they're arranged isometrically. And so in HIV, you get a distorted cone. And so you can see idealism 2 here is how people are playing with symmetry and ideas of what would be fitting, and we'll come on to fit in a more general sense in a moment, to come up with theories which turn out to be correct for how it is that nature is ordering itself chemically. And I want to go on to observation, and this relates directly to what Milena was talking about. I want to say, first of all, something about poo sticks on the left-hand side. So poo sticks is here because I think that many experiments, if not all experiments, have a close relationship to play. And play, from my point of view, is an aesthetic experience. It's something that we enjoy and have delight in. And the reason why I choose poo sticks is because many of the kinds of experiments that I would do or colleagues of mine would do involve taking our sample we want to study, placing it in some kind of medium and exerting some kind of force on it, such as, for example, Milena referred to centrifugation used in the Mesos and the Stahl experiment. What they did was that they centrifuged the DNA to separate it differentially depending on how dense it was. And that is placing their DNA in water and gravitationally spinning it. And then you get movement of the thing and you follow how it moves. And that tells you something about the structure of the DNA. Poo sticks is relevant because many of the systems that we study involve taking our system, placing it in a medium of different kind and applying force to it. And that is very playful, actually, and it feels very playful. There's an enjoyment in grappling with the experiment. And so that's why poo sticks is a good analogy. The diamond light source down the road at the Harwell Research Campus, Milena also referred to the much larger experimental machinery we now use. The diamond light source is an example of one such system. It's a very bright source of tunable X-rays. It's how you would determine structures like the virus structure I showed in the previous slide. And again, there is delight in the design of huge machines with highly sophisticated physics behind them, which enable you to achieve the biological question, answering the biological questions you wish to. But we can observe more directly, and you can see a movie down here taken using a technique called atomic force microscopy of a conformational change occurring in a protein that we work on, as it, in this case, attacks bacteria. And you can see that there's a clockwise movement as it undergoes its conformational change, which is a brightening of the whole structure. So there's delight in observation as well, which I think feeds into our aesthetic experience in science. Another way in which aesthetics comes into the kind of data and nature area that I want to talk about, and particularly in biophysics, is that we can represent our systems in ways which people commonly will find aesthetically pleasing. We can depict structures of proteins like this perforin protein forming a pore, depict them as attractive ribbons. We can color them nicely. In a trivial sense, this is aesthetic, but people do kind of like it. They do warm to the representations that you can give. They will even sometimes ask you, are they really that color? And of course, they're not really that color. It's a way that enables us to try and depict an idea that we want to come across with. But this is pulling together a whole load of data we've got into a movie, which enables us to explain ourselves. Representation is another form of aesthetic process, in this case, aesthetic kind of discourse, if you like, in biophysics. And then form kind of pervades the issue of how we see beauty in nature and data. The form of an alpha helix on the left-hand side there, the form of in this case, the SARS-CoV-2 spike, which is the bit of the protein that attacks our ACE2 proteins on human cells in order to insert into them. The forms which we might be able to relate to other forms in the world. So colleagues of mine studying the SARS-CoV-2 spike noticed that the receptor binding domain bore a resemblance to a torso, such as you might find in a Greek statue. And so that enabled them to organize their thinking as to where antibodies were binding and where the more powerful antibodies were binding. So you can see there's a kind of pooling together of different aesthetic senses in order to bring out some general ideas or some take-home messages that people wish others to understand from their work. And it's about the form of the information and how we can display it. And then fit. I said I would mention fit, and this is kind of relates also to things like DNA, where clearly we're talking about the fitting together of two strands. But fit, and this for me was how I really got into a sense of the beauty in science, and really what led me to become a biochemist. The fit of molecules together. This molecule, something called EDTA, bound to a divalent cation, a metal ion, or the fit of drugs to target enzymes we might be trying to inhibit. And that's a structure of Paxlovid, which is a commercially marketed, it's now available in the clinic, inhibitor of the SARS-CoV-2 polymerase. And that is now out there marketed. It's enabling people to better fight off the coronavirus. But what enabled that work to be done was solving structures and then aesthetically engaging with them and building models into those structures in order to understand how we could inhibit the enzyme, and having inhibited, then how we can design more powerful drugs in order to inhibit it better. And that's exactly what that paper describes. And finally, I think uniqueness is something which you find as an aesthetic principle in general. There's the uniqueness of a single rose petal, the uniqueness of anything isolated that grabs our attention in the wider world. But I think in nature and data, in biophysics, there's a sort of uniqueness even to things which show no apparent order that you can easily perceive, no symmetry, but the unique complexity of something like the ribosome and the ways in which the bits of RNA and protein come together to form that structure it seems to me, another way in which aesthetics is experienced by people studying nature with data, as it were. And so I've had these six kind of key ideas: idealism, observation, representation, form, fit, and uniqueness. They are all different ways, practical ways in which I think aesthetics drives or is an experience engaged with by scientists in studying nature with data and doing biophysics. And if I had to group them all together in terms of what is going on, I think that the scientists are recognizing something when they see beauty in their data or find beauty in their experimental, what the experiment is showing them or the structure they determine. And also there is a delight that they have, and this would apply to the experimental process as well as to the actual results that you achieve, and which delight you with their form, with their fit, with their uniqueness, the way they represent a new understanding to you, and with the idealism that they might elicit from you as to how this system might work. And there I'll finish.
I'm not a historian, and I'm not a sociologist, and I'm not a philosopher either. I'm just your average theoretical physicist. And I guess the reason I ended up at these meetings that I wrote a book, which is called Lost in Math. And I apologize to the British part of the audience for the lack of an S. Though, as you can see in the top right corner, the French, for some reason, used the English title in the British spelling. So this book is very much a personal story. It's my own story. I was trying to figure out what criteria I should use to pick a research topic. And my work has mostly been in the foundations of physics, by which I, broadly speaking, mean those areas where we're concerned with the really, really big questions like what is time? How did the universe begin? What is matter made of? That kind of stuff. So all of what I'm about to say is really about the foundations of physics, not about physics in general. As you can see, the subtitle of the book was How Beauty Leads Physics Astray, and writing a popular science book, so this was my first popular science book, has certainly been an interesting experience. And I found out there is a significant fraction of people who think that by reading the title of a book, they can tell what comes in the 300 pages afterwards. A lot of those people got away with the idea that I'm saying scientists shouldn't talk about beauty, talking about beauty is bad, 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 don't do it. Now, I would like to think that the opinion that I expressed in the 300 pages that followed the title was a little bit more nuanced. There are many reasons that scientists talk about beauty, and I'm certainly not saying all of them are bad. Like we've heard many of those reasons already. For example, it's a motivation for them. It's an inspiration. It gets them into science. And I traveled around for my book and I interviewed a lot of physicists, and they brought up all kinds of reasons to me about why they like to talk about beauty. You can find them in the book. I'm going to pick one in particular, which already came up. So this is a quote from Gilbert, who won the Nobel Prize for the quark model. And he said in a TED talk, what is especially striking and remarkable is that in fundamental physics, a beautiful or elegant theory is more likely to be right than a theory that is inelegant. And this might sound really familiar to you now, because if you remember Penrose's quote, which came up in an earlier talk, was pretty much exactly the same. And I could go on like for half an hour with quotes like this. It's a very, very common attitude that you find in the foundations of physics. And I've grown up believing this because everybody says it. And when I say grow up, I mean grow up academically. It's like really common that people tell you this, like that your sense of beauty is a good guide to the development of new models, to our understanding of nature. And to me, it's kind of weird that I stand here and say this today, because it doesn't make any sense. Once you start thinking about it, you begin to wonder, why should it be the case, right? Why can I use my sense of beauty to kind of magically figure out what's a good theory of nature? But I think the thing is that I never thought about it. It's just that so many people told me this, I never questioned it. And there are lots of problems connected to this because, as Ben said in the very beginning, our notion of beauty dictates, to some extent, what kind of questions we even ask, and also what kind of answers we are willing to accept. In the foundations of physics in particular, what's happened is that pretty much everyone uses the same notion of beauty. And that leads them to discard a large number of theories, which is really a problem. And what's happening there is that we use this notion of beauty, which we have learned to identify in successful theories, and then we try to use it to develop new ones. But this keeps us getting stuck with the same type of theories, and it's just not working. And I always refer people to James' book about how the notion of beauty may change with paradigms. So it's a pleasure to finally meet him after I've referred to his book so many times. There are many examples in the history of science and in physics in particular where what's changed in a paradigm shift was actually our notion of beauty. And he already said quantum mechanics is an example, but there's also been the shift from a more mechanical worldview to one based on the notion of fields that extend through space. Or another example that most people are actually familiar with is the shift from cyclical orbit for the planets to elliptical orbits. And in all those cases, at the time, people thought that the new thing is really ugly. But today, physicists just accept it. It's just the way that it is. And I don't think they think about it as ugly. And so what happens if we insist on a particular type of beauty in the development of new models is that we get stuck. So in theoretical physics, our models are very, very mathematical, and the type of beauty that theoretical physicists talk about is correspondingly basically mathematics. And I'm not going to explain exactly what I mean by those three hidden rules of physics, as I call them. They are basically technical mathematical requirements that people use in theory development, and they're hypotheses, but their status as a hypothesis has become forgotten. And that's the problem. I think this is why we've gotten stuck. I should maybe say that this notion of simplicity, which physicists use, is very strongly based on symmetry. So it probably has an evolutionary origin to some extent. We just find symmetry pleasing to look at. We see this in the development of new models in the foundations of physics. And that has consequences. There are lots of theories and predictions that have come out of theoretical physics based on arguments from beauty, based on asking certain questions about the theories that we currently use. They have perceived shortcomings because they're not beautiful enough. And there are lots of particles that physicists have proposed to remedy this perceived lack of beauty in the theories that we have. Axion supersymmetry, string theory, you've probably heard of this. WIMPs is a particular type of dark matter, and lots of other things that physicists have looked for in dozens and dozens of experiments, and they haven't found anything. And I think, well, that's a problem. So why do they do this? I still find this perplexing, I have to say, because it's once you start thinking about it, it doesn't make any sense. Like, why would you expect your personal notion of beauty to be a good guide to the development of new theories? And I think there are a number of reasons for this. One is social reinforcement. I already talked about this briefly in the beginning. It's just you hear it so often and no one questions it, that you just accept it as true, like the same way that I accepted it as true. There's also a certain lack of philosophical grounding, because if you actually were aware that it's a metaphysical requirement that has no backup in evidence, then you might question it more readily. There's also a lot of cherry picking history. If you think of these quotes, which I mentioned, like the one from Gell-Mann and from Penrose, and you could add many others. Feynman already came up. There's also Paul Dirac. You could add him, and a lot of other people that you can add there. Frank Wilczek, for example, has written an entire book about beauty in the foundations of physics. What do all those people have in common, aside from being men? They've all won a Nobel Prize. So there's a certain selection bias. We only take note of the people who have been successful and who then attribute their success to their sense of beauty. But in the history of physics, there have been lots of theories that were once considered beautiful that just didn't work. And on the other hand, there are lots of ugly theories that actually do work quite well. And finally, there are lots of cognitive biases. For example, they're just some people who I interviewed just said, well, if it's not beautiful, I don't want to work on it. And I mean, who am I to blame them, right? But this is a very strong selection for the kind of theory that we work on. Okay, so I don't know about you, but I would certainly like to see progress in the foundations of physics in my lifetime, which brings up the question, well, what to do? But I think the most basic thing we can do is to increase awareness among working scientists. I think that to be a good scientist, one needs some a certain amount of knowledge about the history of science, the sociology, and the philosophy, which is why I think that events like this are really, really important. Thank you for your attention.